The human body contains a fluid volume more than 3 times the size of the cardiovascular and lymphatic systems combined. Historically, textbooks define the spaces holding this fluid at a strictly microscopic scale, intercellular gaps measuring less than 1 micron. The definition occasionally expands to include pericapillary spaces, around 10 microns wide, where nutrient and waste exchange occur. Medical science has operated on a specific assumption regarding these fluid compartments, that they are discontinuous, highly localized, and sealed within the capsules of individual organs. However, multi-organ decellularization techniques reveal an unbroken extracellular matrix spanning the entire body, ignoring the presumed borders of the heart, liver, and gut. Over a century ago, the anatomist Franklin Mall demonstrated that continuous reticular networks exist within the portal tract stroma of the liver. We also see clinical anomalies that defy isolated organ boundaries, like the highly irregular, seemingly discontinuous spread of certain cancers far from their primary tumors. We are going to examine new orthogonal histological data that challenges the boundaries of this isolated organ model. Researchers evaluated the continuity of these spaces using 2 distinct methodologies: tracking the physical movement of inert particles through live tissue and molecularly mapping the structural matrix itself. This suggests that the classical model of isolated, encapsulated organs may be missing a massive body-wide fluid transport system, one that operates in parallel to the blood and lymph. To prove functional fluid flow across tissue boundaries, researchers tracked the movement of non-biological inert particles injected directly into living tissue. The study utilized dermal cosmetic tattoos and topically applied colloidal silver as models for the skin. For internal organs, they examined colon resection specimens containing endoscopically injected India ink. Pigment particles were identified deep in the subcutaneous fascia and mesenteric fascia, far beyond their classical tissue compartments. This histological section of the dermis shows the widespread distribution of both intracellular and extracellular pigment particles, extending from the papillary dermis down into the subcutaneous fascia. Here in the colon wall, we see pigment particles breaching the muscularis propria and settling in the deep mesenteric fascia. A biological counterargument to passive fluid flow is that these particles could simply have been engulfed and physically carried across tissue boundaries by migrating macrophages. If transport was purely cell-mediated, the resulting size distribution of the deposited particles would be entirely random, regardless of how far the macrophage traveled. To test this, researchers physically measured the diameter of the extracellular particles across the different tissue layers. This chart displays particle size distribution moving deeper into tissue. We see a strict size cascade. Mean particle size drops from 0.64 microns in the superficial submucosa to 0.53 in the muscularis propria, reaching 0.41 microns in the deep mesenteric fascia. This mathematically significant size gradient indicates particles move via passive pressure-driven fluid flow through a porous filtering matrix, rather than by random cellular carriage. With fluid flow confirmed, we need to examine the structural roadmap that allows this liquid to bypass rigid organ boundaries. Standard hematoxylin and eosin stains wash away specific matrix components during tissue preparation, causing interstitial spaces to appear as empty white gaps on a slide. To reveal the true structure, researchers used a targeted molecular marker, hyaluronic acid, or HA. HA is a macromolecular component of the interstitium with physical properties that regulate the flow of fluid and solutes through these spaces. The team utilized a multiplex chromogenic assay to map HA alongside key structural proteins like vimentin and CD34. These colon sections contrast empty white H&E artifacts on the left with the actual structure, dense, continuous brown HA staining on the right. This staining traces an explicit anatomical connection from the lamina propria, threading directly between muscle bundles, bridging straight into the mesenteric fascia. This skin section confirms structural continuity is mirrored superficially, highlighting an unbroken brown HA pathway bridging the epidermal intercellular spaces down through the reticular dermis. At its deepest connection, the flow enters the pericapillary spaces directly between the subcutaneous adipocytes. This infrastructure extends systemically, The fibrous adventitia surrounding blood vessels and the perineurium surrounding nerves are densely packed with these continuous HA channels. The empty fixation artifacts of classical histology actually represent a dense, continuous HA-lined anatomical highway. It utilizes blood vessels and nerves to physically bridge entirely separate organs. Particle tracking proves dynamic fluid flow, and HA mapping provides the precise physical infrastructure facilitating it. The classical oncology model assumes that cancer cells must utilize destructive matrix metalloproteinases to break down physical tissue barriers and metastasize. Clinical observations frequently defy this model. Melanoma in transit metastases or the peribiliary spread of cholangiocarcinoma occur without lymph-vascular invasion or visible tissue destruction. When an artery and a nerve pass through an impenetrable organ wall, HA-lined interstitial channels wrapping around them create an open bypass tunnel. Malignant cells slip into this network, entirely bypassing the barrier by traveling along the artery's exterior sheath. Histological evidence supports this mechanism. Tumor nests found deep in these channels exhibit no desmoplastic reaction, proving the cells have not elaborated tissue-destructive digestive enzymes. Cancer invasion, at least in its initial steps, relies heavily on trafficking through these pre-existing fluid pathways, rather than forcefully remodeling tissue. Metastasis does not always require destroying organ boundaries. It frequently exploits this pre-existing non-destructive fluid infrastructure as a path of least resistance. Beyond oncology, this continuous network model directly applies to the rapid spread of infectious diseases. It explains the fulminant pathophysiology of conditions like necrotizing fasciitis. Virulent bacterial strains utilize these widespread fascial spaces to cause rapid necrosis, and can even track along these pathways to cross the blood-brain barrier. This exact structural mechanism also underpins the gut-liver and gut-brain axes. The adventitia of the portal vein and the perineurium of the peripheral nervous system provide direct, uninterrupted pathways for gut microbiome translocation. The scale of this interconnected fluid volume is massive, more than 3 times the size of the cardiovascular and lymphatic systems combined. The fibrous coverings of nerves and blood vessels have no discrete separation from the organs they travel through. The human body operates with an unrecognized third circulation system. Its existence requires a re-evaluation of physiological communication, systemic infection, and the strict definition of an organ boundary.